Welcome to the Explainer for May 9th, 2026. Today, we're diving into the fascinating 2014 paper by Hermans and colleagues titled Dynamic Adaptation of Large-Scale Brain Networks in Response to Acute Stressors, published in Trends in Neurosciences. We're going to explore exactly how our brains literally rewire themselves in real time the moment we're faced with a threat.
You know, the fundamental premise here is that stress completely alters our brain's resources to keep us alive. It's a true double-edged sword. As the authors explicitly state in the paper, stress is a double-edged sword. It causes us to have difficulty focusing our attention, retrieving information from memory, and making decisions that require complex thought. Yet the acute stress response also enables us to rapidly detect threats. It's an evolutionary trade-off. And today we're unpacking the exact, incredibly precise mechanisms that make this happen.
All right, to do that, we'll follow the exact timeline of a stress response. Here's our agenda. 1. Introduction to stress adaptation. 2. The immediate stress response. 3. Experimental evidence and effects. 4. Corticosteroids and cognitive recovery. 5. Long-term network reallocation. And finally, 6. When stress becomes pathological.
Okay, Section 1. Introduction to stress adaptation. Let's start by actually mapping out the specific brain networks that dictate how we respond to acute stressors.
So, to really grasp this paper, we absolutely have to define the two primary competing systems at play here. First up, we have the salience network. Think of this as your brain's internal alarm bell. It's heavily driven by the amygdala and it integrates autonomic and neuroendocrine control to prioritize threat detection and rapid reflexive action. Now, competing directly against it is the executive control network. You can think of this one as your brain's CEO. This system is driven by the dorsolateral prefrontal cortex, the DLPFC. It's responsible for a higher-order cognitive focus, things like working memory, complex decision-making, and goal-directed attention.
And this functional topography perfectly illustrates that stark anatomical divide. You've got the rapid action salience regions highlighted in red and the deliberate cognitive control regions in blue. Under normal, everyday conditions, these networks maintain a nice, steady balance. But the moment a stressor hits, they start operating reciprocally. Basically, it's a seesaw. When one goes up, the other is forced down. Moving on to Section 2, the immediate stress response. What's truly fascinating is what happens the very millisecond a threat actually appears.
Our bodies don't just panic randomly. They deploy neurochemicals in distinct, orchestrated waves, creating this beautiful timeline of network reallocation. It kicks off at exactly 0 minutes with catecholamines, followed by rapid, non-genomic corticosteroid effects hitting at about 30 to 60 minutes, and then the much slower genomic corticosteroid effects developing 1 to 4 hours later.
Let's look at that 0-minute mark. Boom! Catecholamines, specifically norepinephrine and dopamine, completely flood the system. They bind to specific adrenoceptors and instantly upregulate the amygdala while stripping resources right out of the prefrontal cortex. This rapid chemical release forces a brain region called the locus coeruleus into what's known as tonic mode. Basically, it flips your brain's sensory switch to always on. This massively ramps up your sensory gain. You instantly become hypervigilant, actively scanning for danger, but the trade-off is you temporarily lose your capacity for complex, goal-directed thought.
Section 3. Experimental evidence and effects. It's one thing to theorize about all this network balancing, but how did Hermans and his team actually prove it in the lab?
Well, they operationalized the study by rigorously measuring structural and functional changes using bold fMRI, which tracks blood flow in the brain, right alongside a suite of physiological and behavioral metrics. They monitored heart rate and cortisol to verify the stress response. Then, to test the executive control network, they measured working memory performance using a classic NBAC task. And to test the salience network, they assessed how strongly subjects reacted to emotional facial expressions.
They broke this down into two distinct experimental conditions to really separate the immediate shock of a stressor from the chemical mechanisms of recovery. Experiment 1 compared acute stress induction directly against a control condition to see what happens right at impact. Experiment 2, however, focused on administering hydrocortisone versus a placebo at various time intervals. This was designed to isolate the specific role corticosteroids play in our cognitive recovery.
Check out the effect sizes from Experiment 1. Acute stress literally doubles the activation signal in the amygdala. In the control condition, reactivity to emotional stimuli triggered about a 0.15% signal increase. But right after stress induction, that bold fMRI signal change jumped to roughly 0.30%. The threat detection system is just massively hyperactivated.
And remember that seesaw effect?
As the amygdala fires up, the executive control network is practically shut down. When looking at the DLPFC during a working memory task, the activation plummets from a healthy 0.45% signal change all the way down to just 0.1% under stress. The brain's resources have been completely reallocated away from higher-order thinking.
The fMRI scans provide stunning proof of these functional findings, showing exactly where network activity spikes and drops. You can vividly see the amygdala lighting up during stress, while the DLPFC simultaneously goes dark. It is a brilliant visual representation of our neurobiology prioritizing immediate short-term survival over complex cognition.
All right, section 4, corticosteroids and cognitive recovery. So the threat has passed. How does the brain build back toward cognitive balance? Because staying hypervigilant is exhausting and, honestly, dangerous long-term.
To stop the salience network from just running wild, our brain deploys corticosteroids to gradually shut down that amygdala hyperresponsiveness. If catecholamines are the match that starts the fire, corticosteroids are the heavy blanket thrown on top. They bind to glucocorticoid receptors to actively reverse those intense early effects, slowly but surely pulling the system back toward homeostasis.
Which brings us to section 5, long-term network reallocation. The absolutely crucial takeaway here is that full recovery isn't instant. It requires slow, genomic cellular changes.
In fact, it takes up to 4 hours for the genomic effects of corticosteroids to alter gene transcription and actually rebuild our working memory capacity. These hormones are literally changing protein levels inside your neurons to restore your ability to think clearly and focus on long-term goals.
The data from experiment 2 is wild here. It shows that we only get a massive restorative spike in the executive control network after about 240 minutes. Look at this. The placebo group hovered around a 0.50% signal change. If tested just 30 minutes after hydrocortisone administration, the DLPFC signal actually drops further to about 0.35%. But if you wait 240 minutes, 4 full hours, the signal dramatically surges to roughly 0.75%. The brain is actively overcompensating to get your higher-order cognitive functions back online.
Section 6. When stress becomes pathological. What actually happens when this incredibly intricate, time-dependent balancing act breaks down? The authors point out a major clinical implication: maladaptation. This occurs when the body fails to mount that secondary corticosteroid recovery phase. It might just be the core mechanism behind severe psychiatric conditions like PTSD and burnout. If that genomic counteractivation fails, a person is essentially left stuck at 0 minutes, permanently hypervigilant, constantly anxious, and entirely unable to exert cognitive control over their emotional responses. Which leaves us with this final, rather profound question to ponder. If our brains are beautifully hardwired to dynamically restore balance after an acute threat, what happens to our neural networks when the stress just never stops? It really highlights the absolute necessity of downtime and recovery for our basic neurobiology. Thanks so much for joining me for this explainer. I hope it gave you a fresh, structural understanding of exactly how your brain navigates a stressful world.